caught in the crossfire. It's caught in the crossfire.
Baby, wag ka nang mag-alinlangan pa. Alam mo naman talaga na kailangan ka. Makatulad mo ay
Hi guys! Welcome to our YouTube channel. And what's up mga ka-CHEME? Ka-CHEME sa Biochemistry. So, ayan nga, ipapakilala ko sa inyo ang groupmates ko. Hi groupmates! Hi! So yan, by the way, I am Bernice Jean C. Claveria. And I'm Giselle Gamad. And I am Jesarelah Ulep.
My name is Giselle Gatcho.
I'm Lawrence Julia Simangan.
And we are your ka-CHEME sa Biochemistry. By the way guys, we are here pala to execute our vlog sa mga lab experiments namin na pinapagawa ng aming magandang prof, Ma'am Jinky. So, we are here to be your guide. Charot! And ginagawa sa tao na
Drop it.
Diffusion is the net movement of molecules or ions from region of higher solute concentration to a region of low solute concentration. In biochemistry, diffusion allows the movement of essential solutes in and out of the cell membrane. It is said that in higher temperature, diffusion rate increases because the kinetic energy of solute molecules also increases in its movement in the solution. Let us compare diffusion rate among two variations of temperature. First, fill two 250 ml beakers with 100 ml of water each. Next, warm the content of one beaker. Now, place the two beakers side by side and drop at the same time 2 to 3 crystals of potassium permanganate into each beaker. let us observe and compare the rate of diffusion of the solute.
A few moments later. Eventually. Meanwhile.
Osmosis is considered as the counterpart of diffusion. It is the movement of solvent particles across a region of dilute or lower solute concentration to a saturated or higher solute concentration until the concentration of the two regions reaches equilibrium. Cells uses osmosis as a way to maintain its internal environment and react to changes in its external environment. Let us try to simulate osmosis. Let us start by filling the bowl of the tube with 30% sugar solution until it reaches 10 cm in the tissue tube stem. Make sure that you cap the stem of the tissue tube with a finger. Next, cover the large end of the tissue tube with cellophane and tie it using a rubber band. Turn the tissue tube upside down and suspend the tissue tube in a 500 ml beaker with distilled water. See to it that the level of the sugar solution coincides with that of the water outside the tissue tube. Now, let us measure with a ruler the level of the sugar solution every 30 minutes for 2 hours. Now, let us proceed to the third membrane transport process, dialysis. Dialysis is the process of separating molecules on the basis of their size in a semi-permeable membrane. It applies the force produced by diffusion and the filtering capacity of a membrane. Cells uses dialysis because cell membrane contains pores, also called membrane proteins, which limits large molecules to pass through, including proteins. Let us simulate dialysis that is being used by the cells, and afterwards, let us test for the presence of proteins and any other large molecules. And in this case, let us use milk as the sample. Let us start by soaking 1 square foot of cellophane in water for a few minutes. Afterwards, fold it like a filter paper and fit it in a glass funnel. Now, pour 50 ml of milk in it. Collect all the edges of the cellophane and tie the cellophane with a string to secure it. Leave some airspace above the milk. Place the bag into a 500 ml beaker and suspend it so that it almost touches the bottom. Add distilled water to the beaker. Make sure that it is at an equal level with the surface of the milk. For 1 hour, stir the water outside the milk and let the milk dialyze. Dialysis is undergoing if the water outside the milk is turning colloidal or turning white in appearance. After 1 hour, collect the dialysate. let us test for the presence of proteins and sugar in the dialysate. For protein testing, pipette 1 ml of the dialysate and place it in a test tube. Then, add 1 ml of 25% trichloroacetic acid solution. The presence of white precipitate or turbidity will appear if proteins are present. Now, let us test for sugar. Combine 1 ml of Phelix A and 1 ml of Phelix B in a test tube and heat it in a boiling water bath. Add 1 ml of the dialysate and continue to boil for 1 to 2 minutes. A yellow or red precipitate indicates the presence of sugar.
So what's up mga ka-CHEME? Have you learned something on our experiment today?
See you in our next experimental journey.
Thank you for watching! Bye-bye!
Bye-bye!
Like
And subscribe!
Bakit tumuturo ka dito?